Hello everyone. In last session, we have seen about the basics and fundamentals of animal cell culture. In today's session, we will see about the types of animal cell culture. In general, there are two types of animal cell culture. Number one, primary cell culture, and number two, secondary cell culture. We will start with primary cell culture. In primary cell culture, the cells are derived from the tissues directly by physical or chemical disintegration methods or enzyme digestion methods. Here, the cells are induced to grow in plastic or glass containers with complex media. The cell growth rate is very low here and they are heterogeneous in nature. However, we prefer primary cell culture over cell lines because they are the most representative form of the cells of the tissue that they are derived from. The morphological structures of the cells of the culture are usually of four types. One is epithelial cell type, another one is epithelioid cell type, the third one is fibroblast cell type, and the fourth one is connective tissue cell type. In epithelial cell type, the cells are polygonal in shape. They appear flattened as they attach with the substrate and form a thin continuous layer. They form a monolayer in surface. In epithelioid cell type, they have a round outline and they do not form sheets like epithelial cells. In fact, they do not attach with the substrate. In fibroblast cell type, the cells are angular in shape and are elongated. They are bipolar or multipolar. They form open rather than tightly packed cells. They attach to the substrate. In connective tissue type cells, the cells are derived from the connective tissues such as fibers, cartilage and bone. They are characterized by a large volume of fibrous and amorphous extracellular materials. Primary cell culture has its own advantages and disadvantages. Advantages are they represent the best experimental model for in vivo studies. Next one is they share the common karyotype and characteristics of the parent tissue, which is not the case in cell lines. The disadvantages are they are very difficult to obtain, they have a very short lifespan, and they are easily getting contaminated by bacteria and virus. Now we will see about the secondary cell culture. When primary cell culture is passaged or subculture for long period in fresh medium, they form secondary cell culture. Secondary cell culture is long lasting due to the availability of fresh nutrients at regular interval. The passaging or subculturing is done by enzyme digestion of the adherents. Then it is followed by washing and resuspending of the required amount of cells with appropriate amount of media. When doing secondary cell culture, the subculturing or passaging is done with enzymatic digestion of the adherent cells. This process is called trypsinization. In trypsinization, we rinse monolayer culture, add trypsin EDTA, incubate, stop trypsinization, spin the cells, add fresh media, count the cells, seed a new flask and incubate at 37 degrees Celsius. Like primary culture, secondary cell culture also has its advantages and disadvantages. Advantages are they are easily growing and they are readily available. We can obtain large population of similar cells that can be transformed to grow indefinitely. In culture, they express and maintain cellular characteristics, and these secondary cell lines are useful in virological, immunological and toxicological research. Disadvantages are that these cells have tendency to differentiate over a time of culture and they may generate aberrant cells. Now we will see about cell line or cell strain. What is cell line? Primary culture, when subcultured, can become cell line or cell strain that can be finite or continuous depending on the lifespan in the culture. So, according to the lifespan in the culture, it can be divided into two types. One is finite cell line, another one is indefinite cell line. Now, we see about finite cell line. What is finite cell line? Cell lines with a limited number of generation or growth is called finite cell line. They are slow growing, that is 24 to 96 hours. They are characterized by anchorage dependence and limited density. Now, indefinite cell line, what are they? Indefinite cell lines are obtained from transformed in vitro cells and cancer cells, and they can be cultured as a monolayer culture or in suspension form. They grow rapidly with a generation time of 12 to 14 hours, and they have the property to be subcultured indefinitely. They may exhibit aneuploidy or heteroploidy due to the changes in the chromosome numbers. Immortalized cells are the transformed cells with altered growth properties. The classic example is HeLa. HeLa cells are the classic example for these immortalized cells. They are obtained from the cervical carcinoma transformed by human papillomavirus. So these indefinite cells are easy to grow, maintain and manipulate. However, these cells have a tendency to differentiate over the period of time in culture. Now we will see about some general points related to commonly used cell lines. Nowadays, mammalian cell culture is a prerequisite for the production of biologically active substances in an industrial scale. With advancements in animal cell culture technology, a variety of cells are used to produce vaccines, therapeutic proteins, anti-cancer agents and pharmaceutical proteins. The cells from human, animal and insect can be used to produce cell line. A cell lines that can be grown in suspension culture are preferred because suspension cells have a good faster rate. And when we choose a cell line, we have to consider certain parameters. The parameters include growth characteristics, population doubling time, plating efficiency, saturation density, growth fraction and the ability of the cells to grow in suspension culture. Chinese hamster ovary cells, shortly CHO cells, A549 cells, Vero cells and MCF cells are the commonly used cell lines. Now we will see about the advantages and disadvantages of this indefinite or continuous cell lines. Advantages are they are easily to be grown and maintained in the culture. They grow very fast and we can achieve very high cell density. Indeed, we can obtain large population of similar cells. And doing gene manipulation is easy in these indefinite cell lines. Serum free and protein free medium is available for widely used cell lines. And moreover, the cell lines in these indefinite cells are capable of growing in the suspensions in large scale bioreactors.